The overall goal of this procedure is to prepare Arabidopsis ovules for whole mount hybridization and immunostaining. This is accomplished by first fixing fresh flower buds in the fixative solution. The second step is to carefully dissect and embed ovules in miniature acrylamide pads directly on microscopy slides. Next, tissue processing enables tissue clarification and permeabilization. The final step is to incubate the treated samples with an antibody solution for immunostaining or a labeled probe for fluorescent in situ hybridization. Ultimately, high-resolution confocal imaging followed by three-dimensional reconstruction allows for quantitative analyses in whole mount at the single-cell level.
This method was initially employed to provide insight into chromatin organization in the ovule, but it can also be applied to analyze other cellular compartments in other tissues of the plant and in other model organisms.
Into microfuge tubes filled with freshly made BBO buffer, chilled on ice, add 20 to 30 carpels per tube. Set the tubes to rock gently at room temperature for half an hour. Next, spin down the tubes for 1 minute at 400 Gs. Then, carefully aspirate the supernatants, discard them, and add a milliliter of PBT to the carpels. Place the tubes on ice. For each tube of carpels to mount, have prepared on ice 5 tubes containing 200 microliters of freshly made 5% acrylamide mix